differentiation, de-differentiation and re-differentiation. Differentiation, here after the production of new cells from the meristematic tissues, here the cells are undergoing elongation and differentiation. Differentiation means here the new cells, here they are converting into the mature cells, and the mature cells are generally they are involving in one certain function. So they are assigned to one function. Here the differentiation we can write like this, here the primary meristems. The primary meristems. The primary meristem is a root apical meristem, shoot apical meristem. So these are the here primary meristems. So primary meristem is nothing but it is present from the beginning. Here the primary meristem, it is producing the here new tissues or new cells. The new cells are here differentiated, so matured, and they are involving in the one certain function. Here the produced cells, they are not having the meristematic activity. The meristematic cell or tissue, it is producing here the new cells. Some cells are becoming as permanent cells. Here it is a permanent cell. Okay, here it is meristematic cell or meristematic tissue. It is dividing some cells, and some cells are, all these cells are not having the ability to produce the new cells. Only here a few cells are having the capability to divide, and some cells become as permanent cells. So what is permanent cell? Here they have lost their capacity to divide. Here the produced daughter cells or the young cells not having the, all the cells are not having the meristematic activity. Some cells have lost the capacity to divide. So these cells are called as permanent cells. Here for example, permanent cells are a parenchyma, collenchyma, a sclerenchyma. So all these are primary permanent tissues. Here from meristematic tissue, here the cells are produced, the newly produced cells are differentiated into permanent cells. The permanent cells are not having the ability to produce the new cells. So they are matured, are differentiated and involving in certain function. So they are permanent cells. Here examples for permanent cells: parenchyma, collenchyma and sclerenchyma. So it is a differentiation. So by differentiation method, and next one is de-differentiation. Here the de-differentiation is a completely reverse to differentiation. Here de-differentiation is nothing but from primary permanent tissue. Here primary permanent tissue is not having the ability to divide, but when the plant or organ or organism needs, based on the requirement, the permanent cells again regaining their capacity, regaining the ability to divide. So the primary permanent tissues are regaining the ability to divide. It is called as de-differentiation. By differentiation method, the permanent tissues are produced from the meristematic tissues. Here like this, they are involving in the some functions. Here tracheary elements also involving in some function like water conduction, tracheary elements. So during the differentiation method, here the cell walls are thickened, protoplasm, here in this case the protoplasm is lost, so and involving in the some functions. But here in the de-differentiation method, these cells, so which have lost the capacity to divide, these cells, permanent cells, primary permanent cells, regaining their ability to divide. That is, so they are converting into the meristematic tissue. Permanent tissue is converting into the meristematic tissue. So this meristematic tissue is not present from the beginning. So this meristematic tissue is newly developed. So the newly produced meristematic tissue, it is called as a secondary meristem. Primary meristem is present from the beginning. Secondary meristem is newly formed from permanent tissue, primary permanent tissue. So for example, here secondary meristems, interfascicular cambium, interfascicular cambium and cork cambium, interfascicular. Within the vascular bundles, intrafascicular cambium is present, that is primary. But here interfascicular cambium. So if you observe the vascular bundles, here it is intrafascicular cambium, that is primary. But here what we are discussing here, secondary meristem. Secondary meristem is produced between the vascular bundles here. First starting here the secondary, this interfascicular cambium. Interfascicular cambium is absent but newly formed. Newly formed. So the newly formed secondary meristem, it is from permanent tissue. Here parenchyma is present, or medullary rays are present, parenchyma. So parenchymatic cells are permanent cells converting into the meristematic cells. So it is interfascicular cambium, it is secondary. And outside the stele also here cork cambium is produced, cork cambium. So the cork cambium is what is cork cambium. Cork cambium is also not present from the beginning, it is also newly formed. So these meristematic tissues are cambial tissues. So these lateral meristems, here they are the secondary meristems. Primary meristem is present from the beginning, secondary meristem is newly formed from the permanent tissue. So the formation of meristematic tissues from the permanent tissue, it is called as de-differentiation. And third one is re-differentiation. Re-differentiation is it is also equal to differentiation. Again differentiating, re-differentiation. Re-differentiation means here the permanent tissues are produced from the meristematic tissues. And here also it is differentiation, like a differentiation. Here also permanent tissues are produced from the meristematic tissue. The permanent tissue is here the meristematic tissue, secondary meristem. Secondary meristem is producing, it is having the ability to divide and producing the new cells. So the new cells, newly produced cells become as permanent. So that is called as secondary permanent tissue. Meristematic tissues producing the permanent cells. So these have no capacity to divide. Here also secondary meristem is also producing the some daughter cells, new cells or tissues. So these cells are not having the ability to divide. They have lost the capacity to divide. So that's why it is secondary permanent. Produced from the secondary meristem. That's why it is called as secondary permanent tissue. Here also permanent tissue from meristematic tissue. Permanent tissue from meristematic tissue. Primary permanent tissue is from primary meristem. Secondary permanent tissue is from secondary meristem. Some examples for here de-differentiation, some secondary permanent tissues. The secondary permanent tissues are produced from the secondary meristematic tissue. So what are the secondary meristematic tissues? Interfascicular cambium and cork cambium. So interfascicular cambium during the secondary growth, here it is producing the xylem and phloem, outside phloem and inside xylem. So phloem, so phloem and xylem are produced by the interfascicular cambium. Phloem is outside the cambium and xylem is inside the cambium. Next, cork cambium. Here it is cork cambium, outside cork and inside secondary cortex. All these are secondary tissues only. So that's why here we can write a secondary. It is also secondary phloem. Here it is also secondary xylem. Secondary phloem, secondary xylem, cork, secondary cortex. So all these are secondary permanent tissues. Here differentiation, de-differentiation, re-differentiation. So differentiation is the meristematic cells are producing the new cells, they have lost the capacity to divide. Here also same. But how the meristematic tissue, secondary meristematic tissue is produced? It is produced from the permanent tissues. Sometimes the plant or organ, it needs here new meristematic tissues. That's why the permanent cells are regaining the capacity to divide, converting into the meristem. So like this here, the tissues are produced. Primary permanent tissues or secondary permanent tissues are produced. So totally, so all these are important for the growth. Here the permanent tissues are continuously they are added. These permanent tissues are continuously added, and here during the secondary growth also these permanent tissues are here added to the organs. So that is leading to the growth. So it is about here differentiation, de-differentiation and re-differentiation.